Welcome students. Today we are going to start NCERT chapter 10, which is Biomolecules, and today is theory part 1. So today we are going to cover NCERT section 10.1.1 and 10.1.2. This is disclaimer. Now what is the objective of today's lecture? So after attending this lecture, you will be able to understand the topics which we are going to cover today. So today we are going to explain the characteristics of biomolecules like carbohydrates. Now for your reference, I have mapped the NCERT sections which we are going to cover today. So we will be covering 10.1 carbohydrates, 10.1.1 Classification of carbohydrates, 10.1.2 Monosaccharides. Last, we will summarize. Okay, so let's start biomolecules, fundamentals of life. Now look at this image. This image shows you different types of biomolecules. Like these are French fries, which is made up of potatoes, and potatoes have carbs. So we have carbohydrates, we have nucleic acid. In DNA, there is nucleic acid present. We have proteins, which is found in milk, eggs, and we have vitamins, which is found in fruits and vegetables. So we eat all these carbs, vitamins, proteins via meals. Then we get the energy to do our daily work. So I can say that biomolecules, they are basic components of life. Okay, they form the structure of cells and tissues and carry out function like catalysis, transport, storage, and they help in regulation of biological processes. Now every living cell is made up of biomolecules. So they are present everywhere inside the cell, and they are the building blocks of life. Now biomolecules, they are the primary source of energy for all cellular and body activities. So these energy, or I can say this energy, is used for growth, repair, movement, as well as maintaining life processes. Now biomolecules, they can act as hormones and receptors that allows cell to communicate with each other. So hormones, they are chemical messengers, while receptors, they receive these messages and trigger responses. So this communication is crucial for processes like growth, metabolism, and responding to the environment. Now we are going to see the characteristics of biomolecules. So first key feature is that biomolecules, they are mostly organic compounds. Okay, organic compound means they are primarily made of carbon and hydrogen atoms, which form the backbone of these molecules. Now in addition to carbon and hydrogen, biomolecules often contain other elements like oxygen, nitrogen, phosphorus, and sulfur. Another important aspect is that functional groups determine the properties of biomolecules. Now, functional groups, they are specific arrangement of atoms within a molecule that give it unique chemical characteristics. Now, another thing is that biomolecules, they are mostly asymmetric. Asymmetric means they are chiral. Now, what does chirality means? Chirality refers to the property where a molecule cannot be superimposed on its mirror images. Okay, very much similar to your right and left hands. So this asymmetry is important in biological systems. Now look at here. This is a chiral carbon. Okay, if you'll see to this carbon, all different four groups are attached. This is 1, 2, 3 and 4. So this is a chiral carbon which has all different four groups. Okay, now if you see the mirror image of this chiral carbon, this is the mirror image. Now both of them, they are non-superimposable. Suppose if I take this particular compound and put it on this one, okay, so they will be non-superimposed. Okay, so they are non-superimposable on mirror images. Okay, so I can say a carbon which is attached to four different substituents is called a chiral carbon, very much similar to this carbon in which all four substituents are different. Now let us see the idea of building blocks and polymerization. So biomolecules, they are often made up of smaller, simpler units that join together to form more complex structures. So this process of joining smaller units to make a bigger molecule is called as polymerization. Now look at this example. This is exemplar of simpler unit for complex biomolecules. So we have carbohydrates. So the simple form of carbohydrate is a monosaccharide, which is a sugar unit like glucose. Okay. Whereas if I see the simple form of protein is amino acid, which is the basic building block of protein. Okay, so the complex molecules which you can see over here is formed by the polymerization of simpler units. Okay, in case such as in carbohydrates as well as in proteins. Now we will see some important biomolecules. First we will talk about carbohydrates. So carbohydrates, they are body's primary energy source. They are made up of simple sugars, which are small molecules that can be quickly broken down to release energy. Example is glucose and fructose. So glucose, it is the main sugar which is found in our blood and is used by our cells for energy. Whereas fructose is found in fruits and also provides energy. Next, let's talk about proteins. So proteins, they are crucial for the structure, function and regulation of body's tissue and organs. So they help build muscles, repair tissues and carry out many vital processes. If we take examples of protein that include keratin and collagen. So where is keratin found in our body? Can you guess? It is found in hair, nails, which gives them strength. Okay, while collagen, it is found in skin and connective tissues, which provide flexibility and support. Now, moving on to amino acids, they are the building block of proteins. Just like bricks, they are used to build a wall. So amino acids, they join together to form different proteins in our body. Okay, so some examples of amino acids are alanine or arginine. Okay, so they are some important biomolecules, carbohydrates, proteins, amino acids. Now let us talk about enzymes. So enzymes, they are biological catalysts, which means they help speed up the chemical reactions. Now here are some examples of enzymes like amylase and lipase. Moving on to vitamins. Vitamins, they are organic compounds that are vital for metabolism and maintaining good health. So some example of vitamin includes retinol, which is a vitamin A, and thiamine, which is a vitamin B1. So if you see, this is the structure of retinol, and you can see thiamine. Okay, so how this looks like. Now we talk about nucleic acids. So these are molecules that carry genetic information, which means they store and transmit the instructions which is needed for organism to grow, develop and reproduce. So nucleic acids, they are fundamental to all living things. So some example of nucleic acids includes DNA and RNA. So DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. Okay, we will understand each one of them in detail in upcoming lectures. So if you look, this is the structure of RNA and this is the structure of DNA. Okay, so RNA is single stranded and DNA is double stranded. Okay, now let us talk about hormones. So hormones, they are chemical messengers that help regulate various processes in the body like growth, metabolism and mood. So they travel through the bloodstream to target organs and tissues. So examples of hormones include insulin, which help control blood sugar levels, thyroid hormone, which regulates metabolism. Okay. Now we are going to specifically talk about carbohydrates in today's lecture. Okay, so this was just an overview of biomolecules. Now one by one we are going to focus on all the types. So first we are starting with carbohydrates. Now first let's look at the old definition. So carbohydrates, they were once simply called hydrates of carbons. Okay, so this means people thought that they were just carbon atoms combined with water molecules. Okay. Now, if we see the general formula of carbohydrates, this is CXH2OY. So, X over here stands for the number of carbon atoms and Y over here stands for the water units that are present. Okay. If example your X is 6 and Y is 6, that means 6 carbon atoms and 6 water units. So example, if we look at the specific example, glucose. So its formula is C6H12O6. Okay. So if you notice, this fits this pattern. Okay. If you see in the general formula, we have 6 carbon and 6 water molecules. Okay, this would give us 6 hydrogen. This would give us 12 hydrogen and 6 oxygen. Okay, if you put the value of 6 over here, this will be C6 and H2O whole 6. So 6 into 2, 12. So 12 hydrogen and 6 oxygen. Okay. Now, not all carbohydrates fit this formula. Some molecules that behave like carbohydrates, they do not match this pattern. For example, if we take example of rhamnose, then rhamnose do not fix this general formula. Okay. So because of these exceptions, scientists created a new definition. So now carbohydrates, they are defined as optically active polyhydroxyaldehydes or ketones. This means they are molecules with several hydroxyl groups that can either be an aldehyde or ketone group. Okay, and they can rotate the plane polarized light. So they are optically active. So this is the new definition which you need to remember. Optically active polyhydroxy. Polyhydroxy means several OH groups. Aldehydes or ketones, they are called carbohydrates. So if you look at this example, we have this polyhydroxy ketone over here. Okay, so you'll see several OH groups are there. Okay, so this is a polyhydroxy ketone. Okay, and this is a polyhydroxyaldehyde. Aldehyde, how will you identify aldehyde? We have a CHO group. So this is C double bond O and H. And here It can be a R group also or hydrogen also. Okay, and this is a ketone. Okay, you'll see different OH groups are present. So this is new definition of carbohydrates. Now look at the classification of carbohydrates. Okay, so carbohydrates, they are first primarily classified on three types: based on taste, based on hydrolysis, and based on reduction. Based on taste, they are further classified into two categories. One is sugar and one is non-sugar. Now, based on hydrolysis, they are classified into three categories: monosaccharides, oligosaccharides, and polysaccharides. And based on reduction, they are classified again into two categories: reducing and non-reducing. Now, oligosaccharides, they are further classified into three categories: disaccharides, trisaccharides, and tetrasaccharides. Okay. Now, one by one, we will read about all this classification of carbohydrates. Now, first we will see types of carbohydrates based on their taste. First, we'll talk about sugars. So, sugars, they are defined as carbohydrates that taste sweet. This is why things like candy and fruit taste sweet to us. Okay, so they contain these type of carbohydrates. Example is glucose, fructose, galactose. So glucose is found in our blood and is the main source of energy for our cells. Fructose is found in fruits and galactose is a part of a milk sugar. So they are all simple sugars with a sweet taste. Okay, so if you'll see over here, this is an image of a sucrose, which is a common table sugar. Okay, so sucrose is another example of a sweet carbohydrate. Next, we'll talk about non-sugars. So these are carbohydrates that do not taste sweet. So this is an important distinction because not all carbohydrates are sugars. Okay, even though we often use the word carbs to mean sugar, but you need to remember that not all carbohydrates, they are sugar. So if you see the example of non-sugar, it includes starch and cellulose. So starch is found in foods like potatoes and rice, while cellulose is found in plant cell walls and give plants their structure. So both are important for energy and structure, but remember, they do not taste sweet. Okay, so on basis of taste, we have sugars and non-sugars. So if you see this example, this is a typical non-sugar carbohydrate. Okay, so this is a starch, which is a non-sugar. Now we will see types of carbohydrates on the basis of hydrolysis. Okay, first is monosaccharides. So monosaccharides, they are the simplest form of carbohydrates. So if you'll see over here, the word mono means one. So these molecules, they are single sugar units. Okay, you can find their meaning from the name itself. Mono means one. So this is molecules that are single sugar units. So if we see monosaccharides, they cannot be broken down or hydrolyzed into smaller carbohydrate units. So they are already the most basic building blocks consisting of polyhydroxyaldehydes or ketones. Example is of monosaccharides are glucose and fructose. Okay, so both are important energy sources for living organisms. So glucose and fructose, they cannot be hydrolyzed further to give simpler units. Next, we'll talk about oligosaccharides. So oligo over here means few. So these carbohydrates, they are made up of smaller number of monosaccharide units that are linked together. So oligosaccharides, they can be hydrolyzed to yield between 2 and 10 monosaccharides unit. This means that when we add water and break them down, we get a smaller number of simple sugars. Now, depending on how many monosaccharide unit they contain, oligosaccharides can be further classified into different types. Okay, let us look at these subcategories. First is disaccharides. Okay, so disaccharides, they are oligosaccharides which are made up of two monosaccharide units. Okay, example include sucrose, which is a table sugar, and maltose, which is found in malted foods. Next is trisaccharides. Okay, so trisaccharides, see from the name itself, di means two, tri means three. So they are made up of three monosaccharide units. Okay, example raffinose. Then we have tetrasaccharides. So tetrasaccharides, they are made up of four monosaccharides unit. Okay, like stachyose. Okay, it is an example of a tetrasaccharides. So depending on the number of monosaccharides, they are further classified. Di means two monosaccharides unit, tri means three monosaccharides unit, and tetra means four monosaccharides unit. And examples are very important. They are asked in exams. So disaccharides, sucrose, maltose; trisaccharides, raffinose; and tetrasaccharides, stachyose. Now we will see types of oligosaccharides. So disaccharides, they are oligosaccharides that when hydrolyzed produce exactly two monosaccharide unit. Okay. Example is sucrose, maltose, and lactose. Okay. So look at this chemical reaction for the hydrolysis of sucrose. So when sucrose, which is C12H22O11, when sucrose reacts with water in the presence of an acid, it breaks down into glucose and fructose. Okay, so sucrose has this molecular formula, and we have glucose and fructose. Okay, so that means one molecule of glucose and one molecule of fructose. Okay, so look at the reaction. It requires water and acid catalyst. Okay, and the products, if you see, they are simple sugars in their aqueous form. Next, we talk about trisaccharides. So as I told you, trisaccharides, they are oligosaccharides that yield three monosaccharides unit upon hydrolysis. Okay, this means when you break them down, you get three simple sugars. Okay, example of a trisaccharide is a raffinose. So if you look at the chemical reaction, we have raffinose. Okay, when you react it with water in the presence of acid, it breaks down into three units: glucose, 2 units of glucose, and 1 fructose. So they are yielding 3 units of monosaccharides: glucose, 2 molecules of glucose, and 1 fructose. So raffinose, which has molecular formula C18H32O16, is an example of a trisaccharide. Okay, now next we'll talk about polysaccharides. So polysaccharides, they are unique because when they undergo hydrolysis, which is a reaction with water, they break down into larger number of monosaccharides unit. So they yield a large number of monosaccharide units. Now, unlike simple sugars, polysaccharides, they are considered non-sugars. This means they do not taste sweet and they do not dissolve easily in water. Example is starch, cellulose, and glycogen. Okay. So if you look at the chemical reaction, when starch, which has the molecular formula C6H10O5 and N, when it reacts with N molecules of water in the presence of acid, it breaks down into N molecules of glucose. So if you'll see over here, this N represents the number of repeating units in the starch chain. Okay, so this reaction shows how complex carbohydrates can be converted into simple sugars, which are easier for the body to use for the energy. Now we will see types of carbohydrates on the basis of the reduction. First, we will focus on reducing sugars. So notice, not all monosaccharides, which include both aldose and ketose, are considered reducing sugars. Okay, so you have to notice that all monosaccharides, they are reducing sugars that include both aldose and ketose. Now, reducing sugars, they can react with specific chemical solutions such as Fehling solution and Tollens reagent. So, when a reducing sugar is present, these solutions change color or form a precipitate, which is a classic way to test for the sugars in the lab. Okay, so this is an application of their reducing property. So, need to remember that all monosaccharides are reducing sugars and they reduce Fehling solution and Tollens reagent. Now see, all these are examples of reducing sugar. We have glucose, okay, which is a monosaccharide. We have galactose, fructose, ribose, glyceraldehyde, xylose, okay, cellobiose and lactose. So these are all reducing sugars, okay, both in which we have both aldose and ketose. Now next we will talk about non-reducing sugars. So non-reducing sugars, now see what determines if a disaccharide is a reducing sugars. So a disaccharide is considered reducing if it has a free hemiacetal or hemiketal group on its anomeric carbon. Okay, so this is important, which you need to know, that it has hemiacetal and hemiketal on its anomeric carbon. Okay, now how we will define the term anomeric carbon. So anomeric carbon is the carbon atom that comes from the carbonyl group when sugars form a ring structure. Okay, so this is a, you can say, a very key player in the sugar's reactivity. So remember that it's the carbon that was originally part of the aldehyde or ketone group in the open chain. So this carbon is derived from the carbonyl group in the sugar form. Okay, and I hope you all remember a hemiacetal and a hemiketal. So hemiacetal is formed when an aldehyde and alcohol, which is featuring a central carbon with one OH group and one OR group. Okay, and a hemiketal results from the reaction of a ketone and an alcohol with the central carbon bearing one OH group and one OR group. So if you see over here, this is the identification of acetal and hemiacetal. So in hemiacetal, we have a carbon. This is the hemiacetal. So this carbon has one this OH group and one this OR group. So this is a hemiacetal. Okay. And if you see, this is a acetal. In acetal, to a single carbon, both OR groups are attached. So you will see to this carbon, this is one OR group and this is another OR group. So this is hemiacetal, which is derived from aldehyde and alcohol, and this is an acetal. And this is the identification of anomeric carbon. So if you'll see, this is an anomeric carbon, and here also this is an anomeric carbon. So this carbon is being derived from the carbonyls group in a sugar's cyclic form. So you should know all this, that what determines that disaccharide is reducing if it has a free hemiacetal or hemiketal on the anomeric carbon. Now, non-reducing sugars, they do not reduce Fehling solution or Tollens reagent. This means they can't donate electrons to these chemical tests, so they don't produce the color change that reducing sugars do. Okay, so this is a very, you can say, a key way to distinguish between reducing and non-reducing sugars. Non-reducing sugars, they do not reduce Fehling solutions and Tollens reagent. Now we will see the classification of monosaccharides on the basis of functional group. So first notice that if a monosaccharide contains an aldehyde group, we call it an aldose. Okay, so aldehyde carbon is a carbon atom double bonded to oxygen and single bonded to hydrogen atom, which is usually found at the end of the molecule. Okay, so this group is very reactive and influences how sugar behave in chemical reactions. So if it contains an aldehyde group, it is an aldose. And if it monosaccharides contains a keto group, okay, then it is called as a ketose. Okay, so look over here, we have an example of a glucose, which is aldohexose. Okay, so if you'll see that this glucose contains over here aldehyde group. Okay, this is CHO group, which is usually found at the end of the molecule. So if it contains a aldehyde group, then it is known as an aldose. And if it contains a keto group, we see over here it has a keto group, then it is called as ketose. Okay, so this is glucose, which is called aldohexose, and this is fructose, which is called ketohexose. Now moving on further, monosaccharides, they are classified based on two things: number of carbon atoms they have and type of functional groups that are present. So if you'll see over here, look at this table, see the number of carbon atoms and types of functional group, which leads to different categories. If number of carbon atoms are 3, general term is triose. Okay, so in case of aldehyde, it is aldotriose, and in case of ketone, it is called ketotriose. Okay, if carbon atoms are 4, the general term is tetrose. So if it has an aldehyde group, it is called aldotetrose. If it has a ketone functional group, it is called ketotetrose. Similarly, we have 5 carbon atoms, it is called pentose. So containing an aldehyde group, it is called aldopentose, and containing an ketone group, it is called ketopentose. If we have 6 carbon atoms, the general term is hexose. Okay, if it has aldehyde group, it is called aldohexose, and if it has ketone, it is called ketohexose. And finally, we have 7 carbon atoms. The general term is heptose. So if it has aldehyde group, it is called aldoheptose, and if it has ketone group, it is called ketoheptose. Okay, so they are classified on the number of carbon atoms and the functional groups that are present. Okay, now let's summarize. We have what we have studied in today's lecture. So today we discussed about carbohydrates that are optically active, polyhydroxy aldehydes or ketones, or compounds yielding such units on hydrolysis. Okay, we looked about the classification of carbohydrates, which is based on hydrolysis. So how define hydrolysis? It is a chemical process where a compound reacts with water and break down into smaller units. So it is, first we have monosaccharides. So these are the simplest sugars and cannot be broken down into smaller carbohydrate units by hydrolysis. Okay, examples is glucose and fructose. So look over here, mono means one, so they are single sugar units. Then we have oligosaccharides. So oligo means few, so they are made up of few monosaccharides units that are linked together. So these carbohydrates yield 2 to 10 monosaccharides units when hydrolyzed. Example, sucrose and maltose. And next we have polysaccharides. Poly means many. So they are long chains of sugar units. So they are large carbohydrates that give many monosaccharides units upon hydrolysis. So polysaccharides, they are usually not sweet in taste. Example, starch and cellulose. And next we see that it is important to note that all monosaccharides, they are reducing sugars. This means they can reduce chemical reagents like Fehling solutions and Tollens reagent. Okay. So this was it for today's lecture. Thank you students for your attention. I hope this lesson was helpful to you. Thank you.